Hi everyone, my name's Anna. I'm also a scientific training officer in the training team at the EBI. I'm going to be talking today about EMBL EBI resources in general. I'm going to give a few suggestions about ways in which you can access data. I'm going to give you a bit of an idea about what it is that the EMBL EBI does. So to start with, as I mentioned, the aims here really to explain what we do at the EBI, but also to discuss the variety of data that's available. And I think that's probably a big thing that you're all kind of interested to learn about, because the EBI really does provide a really broad range of data resources. So we're going to look at what we have and some ways of accessing those data. What we also want to highlight today as well is further training. So if you learn about a resource that you're interested in today, you also need to learn about the next steps for you. So how you can get training or to discover more about that specific resource. So that's what I'm going to cover today. I'm going to start off talking a little bit about EMBL EBI. So here is exactly where I'm sitting today on a slightly less sunny day, but we're here in the UK near Cambridge. And one of the big reasons you probably know about EMBL EBI is about the data, the public data that we make available through our many resources. And we're also part of the European Molecular Biology Laboratory. And I'm going to talk a little about that in just a moment. But in terms of what we do at EMBL EBI, the main reason I say you're probably aware of us is our data resources that we make available, but we also do have a number of research groups here, and also we make a lot of training available. We provide training courses, so obviously you're here for today's webinar, but we also have a lot of on-demand training that's available for anyone to access anytime. And we run a number of live courses, both here on campus and virtually. And I'm going to talk a little bit about our training later on and show you how you can get access to that training. We also do a fair amount of engagement with industry, trying to bridge a gap between the data resources we provide and many different companies. Through Elixir, we're also involved in the coordination of bioinformatics around Europe. And so those 5 are a very clear main missions. Sorry. As I mentioned, I wanted to talk a little about EMBL as a whole. So we are one of 6 sites at the EBI. So we're the only one in the UK, and we're the only one that is completely computational. The other 5 sites you'll find across Europe. We have 2 that focus on structural biology, so that's Grenoble and Hamburg. In Barcelona, they're looking at tissue biology and disease modeling. And in Rome, epigenetics and neurobiology. We also have the main site in Heidelberg, which includes an imaging center. We have a lot of member states who contribute funding towards the laboratory. And you can find the list here, mostly across Europe, with one exception of Australia. But we are very much a resource that's used around the world. So the services are accessed by people around the world all the time, not just across Europe. We really are there for everyone who wants to use them. And in case you're interested, I got this map, which is a dynamic map that's available on our website. And throughout this presentation, I have a number of links that are within the slides, and I think Ajay has already shared those slides with you, so you can sort of click through these links and follow along if that's what you want to do, or indeed come back to the slides later. So just before I talk about the data resources that we do have at EMBL EBI, I wondered if anyone has any data resources that they know that are provided by the EBI that they're already using. So please do type the name of that resource or resources into the chat if you have been using any. Ensemble is my example here, but I wonder what else other people might be using that you already know about. And then I will go on to tell you lots more that you may not be aware of. The one that I mentioned, Ensemble, I'm sure lots of you have used in some form previously. That's one a lot of people have often heard of, along with Uniprot for looking at proteins that tends to be one that people find us through often first. And one that I'm sure a lot of you have heard of, if not used, is one of the more recent additions to the EBI resource sort of list of resources is AlphaFold. So for predicting protein structures, I'm sure a lot of you have heard lots about that. So as I say, we have many, many different resources. This slide covers lots of the different lists that we have available. There may be some names on this that you may have seen before, and I'm sure lots of new ones as well. So we start off on the left here, looking at chemicals, molecules, and drug discovery. So things like EMBL, I think a few people have mentioned they've used that before for looking at chemicals, open targets, a drug discovery. And there's a few in that area. We also have, as mentioned, ensembles part of our genes, genomes, and RNAs list. And I'm sure many of you have used that before, but there are others such as the European Nucleotide Archive, the ENA, the Expression Atlas for looking at expression profiles, or RNA Central for non-coding RNA. So there's really many in that group. And then moving on to the proteins, the UniProt was the one that I mentioned previously. But we also have, for example, PRIDE for looking at mass spectrometry data for accessing it, the enzyme portal, PFAM for protein families. So there's a lot there as well. One of the newer ones, as well as AlphaFold that I mentioned previously, is the Bioimage Archive. That's got a really broad range of images. So I think if you're working in that area, I really recommend taking a look at the Bioimage Archive to see what kind of thing is available there. And then across the genetic variation and disease data, the newer one here is the COVID-19 data platform, although that has been around for a few years now. The European Genome Phenome Archive, variation data in the EVA. And actually the European Genome Phenome Archive is an interesting one because all of these resources are completely open. The data is all there for you to go on and access anytime you like without having to request access in any way. It's all just openly available, with the one exception of the European Genome Phenome Archive. And that's because it has sensitive patient data. So what you can do is go to the EGA website and you can look at the kind of data that's available. And if you identify something that is useful for you in your research, you can request access. And there's lots of information there about how one requests access to data. So that's the one exception. But other than that, everything is completely open. And then in the final column, we have the literature and knowledge management section. So some of you may have used Europe PMC for accessing articles. Biomodels is one that often is interesting to people because it's quite different. It's looking at mathematical models. And we also have things like Reactome for looking at pathways. So I wanted to give you from this slide the idea that we have many, many different resources, but it's often really difficult to know where to look for your data of interest. So unless you know about a specific resource, you won't know to go to that resource's website. But what we do have is something called EBI search, which allows you to search for a gene or a protein of interest, and then it will help you go to one of these resources based on the kind of data that you're looking for. So that's something I'm going to come back to in a few slides' time. One I just wanted to highlight was our new resource from last year, the pathogens portal that launched in the summer. We actually have a webinar that's happening on the 27th of March. So that's available for you to register now if you're interested, particularly in pathogens and accessing pathogen-related data. This portal has been around for a few months now, so you are able to go and have a look. But if you would rather have a sort of guided tour through it and get some ideas about how to use it, do come along to the webinar in March to get some better ideas. So from this slide, I really wanted to give you an idea about the amount of data. So I've talked about the large numbers of resources, but we're really talking about a huge amount of data here. And you'll notice that the scale on the y-axis here looking at the volume of data is a log scale. So we really are looking at large increases of data. And here's just some of the resources that I mentioned shown on the graph. But if you want to get an idea about the scope of data that is available, the sort of the volume of data, then I recommend taking a look at this article. So every year we have a sort of the EBI in 2023, 2022, and so on, going back through the years, which gives you an idea about the updates that we've had that year, the amount of data, the new resources that are coming through, and that kind of thing. So if that's something you want to sort of learn a bit more about, I recommend this paper here. We're also talking about data for many different species, certainly not just human. It very much depends on the resource that you're looking at. Obviously, some are just human. I mentioned about patient data before, but others cover a huge number of species indeed. And this is a snapshot of just some of those species. The resources are also interlinked. So by that, I mean that if you want to find out a bit about a protein, you might go to UniProt, you find out a bit about that, you can read all about it. But from there, you may want to think about the structure of it. And there are links available on a UniProt protein page that will take you directly to the PDBE, the Protein Data Bank in Europe, to look at structures. And from there, you might move on to one of the imaging resources to see how that structure was sort of identified. So you can end up moving between some of these resources, and there are links available on those resource pages to help you sort of form little workflows to move to find different types of data depending on what the kind of thing you're looking for is. We also have a lot of collaborations between the resources we provide and resources provided by other people. So, for example, the Protein Data Bank in Europe is a really good example in that it's, as the name suggests, it's sort of developed in Europe, so through the EBI, but the data that it is based on is the same data that the Protein Data Bank, which is based in the US, and the PDBJ in Japan is based on. So they're all based on the same data, but the different websites provide some different ways of looking at the data and some different tools alongside it. So if you're already using the PDB, there's no particular reason that you would necessarily want to move to the PDB, but there may be some different ways of looking at data and accessing it that might be suitable for you. However, we have other resources that are standalone and provided solely by the EBI that you can't find in other places. But these collaborations for data resources really sort of help form other collaborations and ensure that we're open and sustainable and making sure that we can keep providing that data for as long as we can. But I'm sure you might be wondering where all this data is coming from. And the answer is very much from people like you, or potentially from you, from researchers all around the world. So you may do some kind of an experiment in the lab, you produce data, and then you can submit it to a resource to make publicly available so that anyone can access it. And you may do this alongside an article. And I'm going to talk a bit more about data submission later on. So that sharing of data kind of fits within this data lifecycle that you may have seen something like in the past, whereby you plan and you collect your data, so your experiments and your data, you analyze it, you do some pre-processing, then you analyze it, you store it, because obviously the data management side of things, you obviously want to make sure that you're keeping hold of your data and you're not losing anything. But you may also then want to share it so that both you and other people can reuse it. And as I say, I'm going to talk a bit about reasons for reuse and for data submission later on. So I mentioned previously about accessing data resources and the fact that there are a huge amount. So we want to be able to give you a place to start that isn't just trying to think up the name of a data resource and searching for it. if you go to the front page of the EBI website, you will find right at the top this big search box, and you can put within that a gene or a protein or a chemical symbol or a name, and just hit search, and we will be able to then provide a sort of a list of data essentially that we have available for that, for example, gene. And you're able to kind of work through it. You can see down the left-hand side, there's all sorts of different types of results. And in a moment, I'm going to give you a bit of a demo about how this works. You also receive these gene and protein summaries, and that really does just what it says. It summarizes information about either that gene or that protein, and provides you with key information, which you can then follow links to gather more information about it if that's what you want. You can then also continue on to a data resource. So you might have been given a little bit of information and you may think, that's what I'm looking for. I need more of that kind of information. And then you can follow it to a specific resource. And this is what I'm going to show you a demo of in a moment. You may also know exactly what resource you're looking for, but don't quite know how to find it. And if that's the case, recommend coming back to that front page and you'll see this find data resource section here. This provides a list of all the different resources, the services and tools available, with a little explanation about what each one is for. And you can also filter based on the kind of category you're looking for. There are a huge number of tools as well as the data resources. So I really recommend having a bit of a flick through this page to see the kind of thing that we do have available. I just wanted to quickly mention as well, we do often allow programmatic access for data resources. It's not necessarily every resource that provides this, but a large number of them do. And each data resource will have information on its website about programmatic access, if that's something that has available. And by doing so, you can automate the pulling of data from these resources into your scripts or your programs that you're creating. And it obviously speeds things up and allows you to work with much larger amounts of data than perhaps you might be able to do if you're manually going to a website and viewing data there or downloading it manually from a website. So programmatic access is available for many of the resources. I'm not going to talk much about that today, but we do have various training about programmatic access available on the training website. So if that's something you're looking for, do go and put that into the search box on the training site to find some more about that. Before I do that little demo, I also just wanted to highlight about citing databases and tools. Each of the resource websites will provide information on how best to cite it. They'll provide a paper in order to do that. So for example, here, this is from the Uniprot website. It has a page that's all about that. But also, one thing I really wanted to highlight was that many resources have data releases that will change the data that's available because there's more and more data being added. And so occasionally the data that you are accessing might change slightly. And therefore, it's really important to make note of the resource data release number, the date that you access data, to make sure that you're able to sort of prove the point at which you accessed it. And if you're able to go back to a previous data release, you would be able to find the data from that day you accessed it, because otherwise you may find that the data has changed since then. But previous data releases are available for you to go back to. So if you've got a record of it, you'll be able to find what you're looking for. So as I mentioned, we're going to have a quick look at EBI search. So hopefully you are following me to the EBI front page. And if that's not the case, I'm sure Ajay will shout out. So on the homepage here of the EBI website, you will hopefully see exactly what I just mentioned, this big search box right at the top of the page. And in here, you can type a genome or protein or chemical, as I mentioned before. You're also able to sort of reduce your search to specific categories if there's something at this stage you're looking for, or you can leave that as all. So hitting search, it then takes me to some results. It first shows me these gene and protein summaries, and I'm going to take a look at the human gene and protein summary for this gene. I'm just going to jump back to the homepage at this point to show you where this find data resource box is here. So this is the one where all data resources are listed. You can see this explore all our data resources and tools page, and that really does list everything. And at this point, I would recommend using the filters to sort of reduce what you might be looking for, but everything is here, so you can kind of get a better idea about what's available. Okay, so jumping back to the gene and protein summary here. This is the gene summary, and you can see that there's lots of information that it's pulled in from Ensembl in this case. And there are links directly to this genes page on Ensembl, so you can follow that straight through, and you don't need to sort of look for the resource. It's all just linked directly from this gene summary. And you'll sort of see similar pages here. So looking at the expression, this is taken from the expression atlas. You see it will take you to the resource there as well. And this is the baseline, but we also have experimental conditions available too. This is an interactive expression map here, and you can explore that. We also have the protein summary page, which is pulling from a variety of different resources. You can see there's Interpro here, which if you look through, you can find out more about protein families. And UniProt as well, which I'm sure a lot of you are familiar with. We're looking at function, disease, gene ontologies, and the sequences. And again, in exactly the same way, you can learn more by coming directly to the UniProt website, and you can see all the different information about this protein that's available down the side, from the disease and variants right through to structures, families. It's all listed on this quite lengthy page. And then when you get down to the bottom of it, you'll also see various references listed too, to learn a bit more in detail. Also just want to move back to the search page. This was after I put in the gene name and just hit search. As you've seen already, an example of the gene and protein summaries, but there's also a huge amount more specific data that you will see down here, going to all the different resources covering so many different areas. You've got protein sequences here, genomes, metagenomes, all the way down to protein expression data. If I keep going, structural data, all sorts of things. And you can use the filter again on the left-hand side to try and find more specifically the thing you're looking for, and then follow on to the resource. So really what I'm highlighting here is, again, the fact that you don't need to know exactly which resource you're looking for. You just need to know the kind of data you're interested in. Pop any gene or protein name into the EBI search on the front page, and then you'll be able to find your way to the right data. Okay, so that was my little demo of EBI search, and I'll come back into the slides now. I briefly mentioned about submitting data. So as we talked about before, a lot of that data comes directly from researchers through them submitting to the resources. In terms of why you may want to do that, it's often really useful a few years down the line in order to remember what's happened to your data. If you've made it publicly available, it's really clear where that data is. It's really well backed up. That's not going to go anywhere. So if you're trying to work out what someone in your group did a few years back and it's been submitted to a publicly available open access data resource, the data's there. You know exactly where it is. It's not going anywhere. It's also great to be able to get some recognition for the data you submit. Often you submit a paper, and to submit the data to a resource alongside that is a great way of people being able to understand exactly what you did, and maybe to reproduce your work or to reproduce your analyses, or indeed to, as I come a bit later, to be able to ask their own questions about your data. So, for example, you may have done an experiment, produce some data for a specific reason to answer a specific research question, but your data may actually be useful to other people to answer slightly different questions. And so by making that available, you're allowing other people to sort of benefit from your data as well. But that may also open the opportunity for collaborations too. So if someone's using your data, they might want to get in contact and understand a bit more about how you produced it, and may want to sort of work with you potentially on further research and further data production. The second one I have there is all about reliability, reproducibility, and it's often the case now that more and more publishers are asking for data to be submitted to a data resource at the same time that a paper is being submitted to their journal, so that they can see the data as they're reviewing it, and they can make sure that everyone who is going to read the paper can also access the data and understand about how it was produced and how it was used and analysed. And it really is making things more open and more reproducible. There's also one I tagged on the end there about there's the potential to be more green, to be more environmentally friendly, because we know how much goes into producing data, and if someone's already done the experiment that you're going to do, maybe all you need to do is sort of take that data that's openly available and use it for themselves, and they have no need to produce it, to generate the data at that point. So there's hopefully a few reasons, but I think we'll see more and more over the next few years journals requesting that data is made openly accessible, and the EBI resources are a way to do that. So in terms of how and where to submit data, if you go back to that front page of the EBI, you will see right next to the Find Data Resources button, there is a Submit Data button. And that will take you to a little data submission wizard. So that will ask you some questions about what kind of data you have, and why you're submitting, talks about why you're submitting to an archive, and it will suggest which resource to submit to. And that will hopefully sort of guide you to be able to submit. On that page, there's also a list of data repositories that take submissions from researchers and the types of data that they take. I wanted to add a quick slide in here about the process of data sharing, because I think one thing that a lot of people don't necessarily realize is the team of biocurators that's often working in the background of a data resource. So for example, if there's a resource that you want to submit your data to, and you carry on with your experiment and you produce your data, you may get to the end of it and find it difficult to submit your data because your metadata, so the data about the data, isn't necessarily complete as the resource requires. And so what I would really recommend is identifying a data resource that you want to submit to as early on in the process of the data generation, so that you can make sure you're keeping track of everything you need to keep track of. You're producing all the data and metadata that you need in order to submit it later on. And what I wanted to really flag here is, if you're unsure at any point of that process of working out what you need and submitting, you talk to one of the biocurators who is available for that resource. And you can do that by contacting the help desk on that resource's website. And there really is a team of people who are ready and willing to help with data submissions. And they really would rather they get questions early on in the process of your submission, rather than sort of trying to help you fix things later on. And so that's a really important thing if you're going to be submitting data. Okay, so I just want to move on to talk a bit about training and support at this moment. So if you head to the EBI training homepage rather than the EBI homepage, you will find that we have these three sections on our front page about live training, on-demand training, and also some support for trainers. And I want to talk mostly about the first two, but I'll mention the third one as well. So in terms of our on-demand training, this is our openly accessible, completely freely available, self-paced learning that's been developed by experts from the EBI, from the wider EMBL, and also from some other sort of experts beyond EMBL. And we tend to cover sort of four different formats. We have online tutorials, which are kind of short introductions to a concept or to a data resource. And the idea is to sort of get you going, get you introduced to that topic. In terms of the resources, they provide introductions, a type of data, how you access it, what you may want to do with it, and also submissions that I've mentioned previously as well. So if you're just getting started with a particular resource, I really recommend coming to the training site and searching for that resource's name, and you'll find an online tutorial about it there. We also have these curated collections. It's the second one in this slide. And these are collections of on-demand training resources on a particular subject. So if you're not quite sure where to start, but you know what topic you're looking for, I recommend taking a look at the collections, and it will help guide you through some of our training that introduces that topic. We also have an introductory bioinformatics collection, which if you're right at the start of your bioinformatics journey, I recommend you taking a look at that one. We have our recorded webinars. So you're obviously one of our live webinars, but they are all recorded, and we make the recordings available along with the slides so that any topic we cover will be included in our sort of section on recorded webinars. We also run live training courses. I mentioned we have them both on site here in Cambridgeshire and we in the UK, and we also have virtual courses. These have a small number of people who attend them, usually around the 35 number of people. So we understand that not a lot of people are actually getting to these courses, but we provide all the course materials. So all the materials that are provided during our live courses are made available, and they are useful for people to learn from, but they're also there for other trainers and teachers, lecturers who want to use our content in their teaching. I mentioned that all this training is completely openly available, and that's very much the case. But you will see as well that there is a sort of functionality to log in and register to the training site. And what that does is allow you to keep track of your learning. So you'll see on courses, you have these mark as favorite and these mark as complete buttons, which will allow you to track things. And you will also have this personalized My Learning page, which will show your progress through our on-demand training. So there's no need, if you just want to access the training, you can do so. But if you would like to keep track of what you're doing, I recommend registering and logging in as you complete some of the on-demand training. One of the other things you're also able to do is to create a playlist if you're logged in. And one of these playlists basically allow you to put together your own list of our on-demand training so that you can create a sort of playlist for yourself to complete, or indeed you can share this playlist with anyone, including anyone who's not logged in. It sort of doesn't matter if they're registered or not. So for example, if you had some colleagues or or you were training or teaching, you may want to share a list of these training courses with them, and you can do so by creating a playlist and just adding some different on-demand training to it. So here's an example that I've made, and if you're following along in the slides, you can try out this link. This is a playlist of our structural bioinformatics training content. and you can sort of work your way through it or take a look and see what that looks like. As I mentioned, we do have people who are using our materials, our training resources to teach others. That is absolutely fine. And we make our resources completely openly available using this CC BY license, which means that you can use it in any way you like. You can modify it. You can sort of take a couple of slides and change them and add them into your own slides, but you just have to say where they come from, and that's all that we ask for you to do if you want to use them. So again, I'm just going to give you a quick demo of the training site to show you a few things around here. So hopefully you've been here before because you came to the webinar. But just to give you a quick idea about what all this looks like, this is the homepage of the EBI training site. Incidentally, if you're on the EBI homepage, you will see these tabs at the top, and the training one will bring you straight to this front page. We have a few featured courses that we like to highlight, and these change fairly frequently. And we also have this live training and on-demand training sections here. So the live training is where you'll find our live webinars, as well as our virtual courses and courses that are based at EMBL EBI. The webinars, as you see, are completely openly available, and our other courses require an application process. Then on demand, we have these 4 different types of training resources that I mentioned before. And here you'll find many recorded webinars from the past couple of years, the online tutorials and the collections that introduce various topics in bioinformatics and the EBI data resources, and the course materials from our live courses. And you can see, because I'm logged in at the top here, I've already marked some of these as favorite or added them to playlists. And if I go straight to my learning page at the top here, I can see everything that I've favorited or everything that I am currently in progress in doing that's listed on my personal My Learning page. So that is largely what I wanted to show you from here. While I am here, I will also just show you the my playlists page here. And this is exactly what I showed you in the slide a moment ago, but you can create your own playlists too and share those with others. Okay, so back into the slides. Right, just a couple more before we finish, and I can see some questions already. So if you need any help at any time, there are various ways of doing so, but you can see here on each of the data resource websites, there'll be a little either help button or a contact us, which will take you to this support and feedback page where you can select the resource that you need some help with in the sort of list that it provides here. Pop in your subject and your question, and someone will come back to you and will give you some help. There are also help pages within each of the resources which provide you with information on how to use them, as well as the training courses that I've mentioned, which provide information about how sort of step by step to move through the different data resources. And briefly, just the paper that I mentioned before, if you want to sort of see an annual review, as it says here, of the highlights of data resource and tools and what's been going on in research here, that this paper is a great way of getting an overview. As AJ mentioned at the start, this is the first of our webinars of 2024. We have many already available for you to register for. I'm sure some of you have already done so. But the start of this year, so February, we are kicking off our Exploring Microbial Ecosystems webinar series. And you can see lots of different topics that we're covering here, along with the pathogens portal, which I mentioned earlier. So these, as I say, are all available to register for now. So please do find them on our website and register. Okay, so I think with that, we will move to questions, and I can probably stop sharing as well.
Great. Thank you, Anna. Thanks a lot for today's webinar. And thanks everyone for joining in and sending in your questions. I think I can start with the first question. It's about, is there any possibility of funding to attend any bioinformatics course that we run?
Yeah, so usually our courses, our live courses don't have any funding that comes along with them. There's occasional exceptions for those, depending on if we're working with sort of another funder to produce the course. But usually it's a case of the individual looking for funding themselves if they get a place in a course.
Yeah, thank you. There's another question around data submission, and I think the question is if somebody has clinical data and they have been advised to submit it to EMBL EBI and some DNA data as well. So how can they approach submitting this data?
Yes, so I think if you go to that data submission wizard page, which I mentioned is linked from the homepage, it'll ask you some questions and then suggest the resource to submit to. Once you've identified which resource is suitable, there will be some information on their website about how to submit that data and what kind of files and what kind of metadata that you need. So that first step is work out which resource it is, but different resources obviously require different information. But as I mentioned, do talk to the BioCurator team. Send a message via the help desk if you need assistance, because they will be able to make sure you're on the right path to submitting, and will be able to break down what it is that you need, and you can sort of go that way. But that's the first step that I recommend. Identify which resource it is, and then you'll find that information that's specific to your file types.
Thank you. There was a question which came in chat. It's about AlphaFold. Do you have a course or tutorial and that tells us how to use AlphaFold database?
Yes. So we have a number of webinars available on AlphaFold. If you go to the training site and you put AlphaFold into the search box, you'll see a few webinars pop up, and they talk a bit about the kind of data available and how to use them, and give you some examples. But we also will be having an online tutorial, so one of the self-paced courses, coming out very soon. So I would keep an eye out for that as well. But the webinars, the recorded webinars, are there already, so check those ones out.
Another question is there an EMBL EBI webinar group and how they can join it?
There isn't a webinar group, but one thing I forgot to mention is that if you register for the website, when you register, it asks if you would like to join our mailing list. And we send out an email about once every 2 months that lists upcoming webinars, new online tutorials, and gives you some ideas about the kind of content that we've added recently. So I really recommend registering to get the mailing list. I think that's really useful. But also keeping an eye on our LinkedIn is a really good way of seeing what we've got coming up. We list all our webinars there, all our series, and that's probably the one of those two ways is the best way to keep up to date.
Okay, a question that came in is about PhD program that's offered by EMBL EBI, how they can apply for it. I mean, I have the link for EMBL International PhD program I can put in chat, but if you have any comment.
Yeah. So the PhD program is offered by EMBL as a whole. So we have a number of PhD students across all the sites, and some of them are hosted here, but it's very much a cohort that's across EMBL, and they sort of work together, and they have training together, and sort of spend time working together. So every year, I think there's actually 2 intakes of PhD students, and these are very much open to anyone from around the world to apply to. You also have postdocs as well, that are based at the EBI. I think there's probably a link very close to, if not the same link as Ajay mentioned for the PhD program. I think there's probably a link on there as well. But we don't have any master's programs. So I can see the questions coming in on the chat. We don't have any master's programs. Occasionally we have interns, but they tend to sort of be a bit after masters, probably mean PhD. But there's absolutely nothing stopping you from approaching group leads to ask about internships. And there's a page again on the website about how to talk to people about internships, but we don't have anything in terms of a master's program as such. And there was a question from someone about future dates for webinars that are focusing on APIs or programmatic access. And whilst we don't have any in the plans at the moment, we do have quite a lot of recorded webinars that focus on those, including one on Uniprot that happened quite recently that was about programmatic access of Uniprot using Python. And there's a really good materials, including a Google Colab, that go along with that. So I think that one's a really good one to sort of start if you're starting to look at programmatic access. That's a good example there.
There's another question in chat. Do we have any training on RNA sequencing?
Yes. So we run a course on this topic, and the course materials are available for that. So the best thing to do in that scenario is to just pop RNA-seq into the search box on the training page, and you'll find a fair amount of content there to have a look through. I also just saw someone asking about they're sort of starting to learn about bioinformatics, and are they looking for things for beginners? Yes. I think I mentioned we have a collection that's called the Introductory Bioinformatics Collection. So go to the on-demand section and tick the collections box, and you'll see that one come up. It does a few things in that introductory collection. It talks about bioinformatics as a topic in general, talks about basic concepts in bioinformatics, and it does that through an online tutorial we call Bioinformatics for the Terrified. So I recommend that. But the collection also includes introductions to data management topics for bioinformatics, which is really important when you're getting started. It talks about metadata and ontologies and things like that. And then it also takes you through some of the EBI data resources, so introduces you to Ensemble and Uniprot, and I think Expression Atlas. So I really recommend that as a place to start, and Ajay has dropped the link into the chat there, so that's a good one to look at.
Okay. What's your view on integration of generative AI and bioinformatic tools and databases?
I think we could do a whole nother webinar on that. Yes, I mean it's definitely becoming a big thing. I think yeah, it's going to become more and more relevant to all parts of bioinformatics, and certainly a lot of our data resources are starting to use AI in a variety of different ways. I think we do have a few webinars that talk about some of these AI. Well, obviously AlphaFold is one that we talked about before. Yeah, I'm not really sure where to start answering that question because there's so much potential, and EMBL as a whole is really focusing on integrating AI in so many different ways. I think there's an article or a couple of articles that are on the EMBL website rather than the EBI website that talk about AI not just in bioinformatics but in biosciences in general, and how many different sort of ways we're starting to use it and already are. So I would definitely recommend having a look at those articles. I'm not sure I can find them quickly, but if you go to embl.org, I think at least one or two of them are on the homepage linked from there. So I think take a look at that.
Yep. There's one interesting comment in the webinar chat. It's about if somebody finds some information missing on our website, how they can move forward and get in touch with somebody to update information.
Yeah, I mean, it depends on which resource it is, but get in contact with them via that help or contact us link that'll be at probably the top right-hand page of the page, sorry. So I really recommend getting into contact with them. They'll be keen to hear what it is that's missing. Some of the resources you're able to add information. So for example, like Uniprot, you can contribute certainly like articles and that sort of thing. But contact the resource and they'll want to hear about it.
Just a new question that came in. I think we still have time. I need help on data analysis of RNA-seq, Go, cake pathway, differentiation, etc.
Yeah, we certainly have training on some of those elements. I would, yeah, pop those into the training site and see what comes out. In terms of pathways, take a look at Reactome, which is a data resource. We have quite a nice online tutorial about Reactome as well, and what you can use it for, and how you can work with the data available from that. So have a look for that one.
Yeah. And we also have a resource about Go analysis as well. You can also have a look.
Yeah, I mean, Go is one of those things that also makes its way into all sorts of training that we have. So, for example, if you're looking at Uniprot, there'll be stuff about that there. But we have specific recorded webinars on Go as well.
Yeah, and just to repeat, as Anna mentioned, if you have any term you're looking for, just put in our search box and look for all the different types of training that we have available. With that, I think there are no more new questions. So I would like to thank Anna again for the webinar today. And I would like to thank everyone for joining in today and making it highly interactive with your questions. So thank you again, and I look forward to most of you in future webinars that we'll be running soon. Okay, thank you. Bye-bye.
Bye.